Good morning, everyone. My name is Alex. I'm a PhD candidate currently in Professor Kushu's lab at UC Berkeley in the Department of Chemistry. And today I'd like to talk to you about my work on tracking fast single molecules using super-resolution microscopy. To start things off, back in 2020, we published a new method called single molecule displacement mapping. And this approach is our recently developed tool that enables us to track quickly diffusing single molecules. It works based off the idea of a conventional storm microscope setup. However, conventional storm microscope setups are unable to track quickly diffusing species, such as proteins in free solution. And really, this is the result of, if you were imagining a single molecule diffusing in solution, that by the time you were able to observe it in the second frame, it would have diffused out of focus within this 10 millisecond time window. In single molecule displacement mapping, or SMDM, we apply tandem pulse illuminations that are closely spaced together, such as 1 millisecond, as shown in this figure here, and this actually enables us to see single molecules moving between a pair of frames. Using the concept of single molecule localization, we're able to then determine the coordinates of the single molecules, and as a result, determine the displacement of the single molecules within this 1 millisecond time window. Now, if we repeat this process, we can accumulate displacement statistics that we can then fit to the diffusion equation to get the diffusion coefficient. One of the unique features of our method is our ability to spatially map diffusivity at nanometer resolution. Because we image in wide fields simply by taking displacements in a select region, such as this magenta versus yellow region, we can locally bin these displacements and fit them to get their diffusivity coefficients. We can use this idea of fitting displacement values into smaller and smaller bins, as shown in this MEOS 3.2 fluorescent protein diffusion in a live CO7 cell, where we use 100 by 100 nanometer size bins in order to bin the displacement and obtain their diffusion coefficients. The color here in this color scale is actually representing the diffusivity of this MEOS 3.2 protein diffusing in the cytosolic regions of the cell. Interestingly, we observe heterogeneity in the diffusivity as we see colors of yellow and green representing diffusivity constants around 25 and slower regions of diffusivity shown by these blue stripes, which are as low as about 10 to 11 micrometers squared per second. So what's really going on here is actually that this MEOS 3.2 protein is actually interacting with the actin cytoskeleton, and as a result of this interaction is slowing down. What this is demonstrating is that we're actually able to spatially map and observe nanometer-sized features and do so using our short time separations. One question that we were interested in investigating was how the protein's net charge impacted diffusivity inside of the cell. Interestingly, as we modified the charge of this MEOS 3.2 protein, we didn't see a significant change in the diffusivity. However, interestingly, when we made the protein positively charged, we saw a significant reduction from about 25 micrometers squared to less than 15 micrometers squared per second in diffusivity, really beckoning the question of what mechanism is driving this diffusivity slowdown. So if we think about the composition of the intracellular environment, there's actually a significant asymmetry in the ions present in the solution, where there's actually much higher degrees of cations compared to anions. As a result, the macromolecules in the solution, such as protein and RNA, tend to balance the charge as a result. What this means for a diffuser, though, is for a negatively charged diffuser, it's only really interacting strongly with the positive ions in solution, whereas the larger macromolecules, such as protein and RNA, the positive diffuser is more strongly interacting with. And as a result of these larger interactions are slowing down much more significantly, we then recapitulated this system in vitro, where we see that indeed charge remains an important determinant in diffusivity value, even in solution. So while negatively charged protein, as shown in these red curves, weren't significantly impacted in diffusivity when we stuck it in a solution of increasingly high amounts of negatively charged protein, the positively charged proteins were significantly impacted in diffusivity and did so in a charge-dependent manner. A second discussion I wanted to mention is my work on testing the limits of SMDM tracking capabilities and doing so in an accurate and reproducible manner. So here we applied SMDM to determine the diffusion coefficients of a variety of different enzymes, ranging from as low as 20 micrometers squared to as high as 100 micrometers squared, demonstrating SMDM's capability of monitoring a wide range of diffusivity coefficients in freely diffusing proteins, and did so in a way that matched theoretical and previously measured reported values. In addition, we showed that SMDM's uncertainty in measurement scaled very well with about 1 over the square root of the number of displacements that we were able to obtain, achieving approximately 3% relative displacement uncertainty with only about 1,000 displacements obtained. And this is a really significant number because we can obtain approximately 1,000 displacements in the span of a few seconds, demonstrating that we can very quickly and accurately determine diffusion coefficients using our single molecule approach. The next question we asked is really how fast can we track with SMDM? And so it turns out actually that using a select set of very bright fluorophores that we're actually able to track anything using these certain fluorophores provided, and can do so in free solution and in an accurate way as shown in this figure here. And this really enabled us to ask the question of how quickly do small molecules potentially move around in the intracellular environment. And so some examples of maybe a small molecule, or molecules as we'll define as less than 1,000 Daltons, could be ATP, glucose, cyclic AMP, all of which have important intracellular functionality, and their transport might be interesting and potentially heterogeneous inside of the intracellular space. What we showed here for this Psi3B molecule is that the diffusion inside of the intracellular environment is indeed heterogeneous, where certain regions, as shown by this black arrow with this in these more yellow regions, were regions of more unhindered diffusion that matched the diffusivity in, similar to the solution-based diffusivity. However, we saw interesting diffusion slowdown also within the intracellular space, perhaps due to slight interactions with membrane structures. We were then interested in asking if there is an intracellular diffusion speed limit for small molecules. Like, how fast can molecules move inside of the intracellular space? So as we know, if we take a single molecule and then stick it inside of the intracellular space, that it indeed slows down, and this is due to the result of the higher viscosity of the intracellular space, as well as the macromolecular crowding. Previous reports had reported diffusivities of approximately 25% of their solution-based diffusivities. However, when we looked into this idea of how quickly single molecules move inside of the intracellular space, we could observe regions of unhindered diffusion that were only approximately about 70% of their in vitro value. And so this is a really significant finding, because what this demonstrates is that actually viscosity is the main driving force in slowing down small molecules and not macromolecular crowding. And this should kind of make sense because macromolecular crowding works off of size exclusion-based principle and viscosity doesn't. So it would make sense that viscosity would be the primary driving factor in reducing diffusivities. And we could really answer this question using SMDM, as we're able to resolve regions of heterogeneous diffusivity using our spatial mapping capabilities. And lastly, I wanted to talk about one of our exciting directions that I'm currently pioneering in my group, which is applying single molecule displacement mapping, but now interrogating the molecular charge of single molecules. What we do here is we apply the same excitation pulses that I showed earlier in single molecule displacement mapping, where we apply pulses between camera frames in order to track the displacements of single molecules, except now we apply an alternating electrophoretic voltage. And if the molecule's charged, it'll be displaced as a result of this electrophoretic voltage. So in this movie, what we can see here is this IgG molecule, and it's kind of dancing in solution. And what's really going on is that due to this application of the electrophoretic voltage, the IgG indeed is charged and is being displaced as a result of this electric field. We can use SMDM in order to characterize how much it was displaced, and we can simultaneously measure the diffusivity as well as the displacement due to the electric field, and as a result, determine the electrophoretic mobility of the single molecule, as well as determine the bulk net charge of the protein. And so in this figure, what I'm showing here is that the IgG protein, we can both identify the isoelectric point of these single molecules as well as identify different charge states as we change the pH of the solution. And we think this is a really exciting direction, as now we have a multidimensional approach in order to simultaneously measure not only the diffusivity of single molecules, but also molecular charge. And with that, I'd like to acknowledge my group and ask if anyone has any questions.